Hi guys, my name is Chris. Excuse me. I attend Columbia University going into my senior year. This summer I was at Hunter College as a SPUR fellow researching this natural compound in green tea called epigallocatechin gallate. Yeah, that's a mouthful. The acronym is actually a lot easier, EGCG. And we studied its effects on astrocyte activation and meth-induced toxicity. So, is meth a serious issue in our country or world for that matter? Yes. Simple answer is yes. Abuse of this illegal psychostimulant has become an international public health concern with an astonishing 15 to 16 million users worldwide. Meth is the most commonly synthesized legal drug in the United States with the highest prevalence in the southern states, Midwestern, and Western states. One of the biggest misconceptions about meth is that it's limited to low-income white males, but that's just not the case anymore. The demographic has shifted to a much broader population that includes women, minorities, and even young professionals. So needless to say, these statistics, among many others, emphasize the dire need for this research on the specific mechanisms of meth toxicity and recovery. Furthermore, research on methamphetamine can not only impart knowledge on the field of drug abuse and recovery, but it can also contribute to knowledge of specific neurodegenerative diseases such as Parkinson's and Alzheimer's. Among the various effects of methamphetamine toxicity, it has been shown that symptoms of Parkinson's arise after abuse. Parkinson's is essentially a neurodegenerative disease wherein dopaminergic neurons progressively degenerate. And methamphetamine has similar pathology. Therefore, gaining a better understanding of certain drugs that act to protect against neurodegeneration and methamphetamine toxicity informs the treatment of certain diseases such as Parkinson's and Alzheimer's. So, what does meth do to the brain? Once past the blood-brain barrier, it has many detrimental effects on the neural environment. As a lipophilic molecule, it can diffuse into the mitochondria and disrupt the electron transport chain. It can displace dopamine and vesicles, releasing them into the synaptic cleft via the dopamine transporter, and it can prevent reuptake of dopamine back into the synaptic terminal from the synaptic cleft. All of these act to accumulate dopamine in the synaptic cleft, which increases postsynaptic stimulation. So while in the synaptic cleft, dopamine undergoes metabolism. This process is called autooxidation. So this process, this breakdown of dopamine produces harmful byproducts called reactive oxygen species. And these reactive oxygen species disrupt the function and structure of proteins, lipids, and sugars. And one of the more downstream effects of meth toxicity includes astrocyte activation, which occurs after these harmful oxygen species are produced. So astrocytes are specialized glial cells that outnumber neurons by 5-fold in the brain. They have many functions, including they create the brain environment, they control the development of neural cells, they control extracellular homeostasis, and for our purposes, they provide for the brain's defense. As the brain's defense mechanism, they respond to specific injuries such as trauma, stroke, infection, and in our case, toxicity, or methamphetamine toxicity. Increasing evidence points towards the potential of reactive astrogliosis, or abnormal astrocyte activation, as having a primary or contributory role in certain central nervous system disorders via loss of normal astrocyte function or gain of abnormal effects. So, once again, finding a drug that counteracts activation of astrocytes could prove to ameliorate the detrimental pathways of meth toxicity. So, in our study, we chose to study the striatum, which is a subcortical part of the forebrain, and it has been proven to be the most significantly affected part of the brain in meth toxicity. So, in meth toxicity, dopaminergic neurons in the substantia nigra are degenerated, and this results in a loss of dopamine in the striatum. So further in the literature, it shows that 25% of striatal neurons are apoptotic 24 hours after meth injection. So this high death rate is the reason for our study of this area, as it seems to be the most affected part of meth toxicity. So, I mean, as I was saying, as a part of the meth toxicity pathology, astrocyte activation is a potential target for certain neuroprotective drugs, or in our case, natural compounds. EGCG is a natural compound found in green tea And recent literature strengthens the perception that diverse molecular signaling pathways participating in the neuroprotective activity of this natural compound renders it a potential agent to reduce the risk of both neurodegenerative disorders as well as meth toxicity. So I mean, it's already known that it enhances the brain's resistance to oxidative stress, but its specific properties go beyond that of just oxygen scavenging and iron chelating. And these properties are what we want to look into, specifically astrocyte activation. So in our experiment, we hypothesized that EGCG would protect against meth-induced toxicity by attenuating activation of astrocytes. So it would reduce the level of astrocyte activation. Astrocyte activation in our study was tested by GFAP levels. GFAP is essentially a protein that astrocytes express. So the higher the level of astrocyte activation, the higher the level of GFAP, and vice versa. So first things first, we had to establish a experimental model. This was to test the proper amount of methamphetamine needed to induce significant damage in the mice. We needed to do this because we were working with a new strain of mice called CD1 male mice that our lab had not previously worked with. So first things first, we created 4 different groups, 3 different concentrations of meth, 30 microliters per kilogram of body weight, 40 microliters and 50 microliters, and then our control was saline. I broke them into 4 groups, sacrificed them 3 days post-injection, and then we performed a tunnel assay on the slides. And what tunnel is essentially an immunostain that binds to damaged DNA. So the brighter the stain, the higher the signal to background ratio, the more damage in the brain. So as you can see, M30 and M50 show the most damage to the brain, but we excluded M50 because there was a high death rate in this group. It was too high of a concentration, so about like 2 or 3 out of 9 mice seized out and died. And if we use M50 for our experiment, it would have probably ruined our statistical significance. So we stuck with M30. And then so to further test the effectiveness of meth on our new strain of mice, we took temperature. So because spike in temperature or hypothermia is associated with methamphetamine toxicity. And as you can see, there's a large spike with M30 and M40, M30 being the largest, compared to saline. So right where the spike is right at the injection time. So we took the temperature 30 minutes prior to injection, and then 30 minutes at 30-minute intervals for the next 8 hours. So this showed us that methamphetamine was actually taking effect and was the reason for the toxicity within the mice striatum. So for our experimental procedure, we broke the mice up into 4 groups again. First group was injected with EGCG and then 30 microliters of methamphetamine. Second group was injected with just EGCG, third just meth, and the fourth just saline. Mice were sacrificed again 3 days post-injection, protein extracted, protein quantified, and then we will be performing Western blot analysis and antibody detection methods next week. So what we expect to find is that the GFAP levels in meth injected with just meth will be much higher than mice injected with both meth and EGCG. So we expect EGCG to reduce the levels of GFAP and thereby reducing the level of astrocytes in our mice striatum. And of course, both the mice groups injected with meth should be higher in GFAP than the mice not injected with meth at all, which is the saline group and just the EGCG group. So I mean, if we did obtain our expected results, this would show us that EGCG does have a significant effect on astrocytes. It does lower astrocyte activation, but what it does not tell us is how this happens. This is something that we want to look into if we do obtain our expected results. I mean, does EGCG have a direct or indirect effect? Does it produce signaling molecules? Does it have a second messenger that affects astrocytes? That's something that we want to look into if we do obtain results. On the other hand, if we do not, we probably go down a different pathway, looking at the other effects of methamphetamine toxicity, such as radical production, lipid peroxidation, etc., and see if EGCG has, how it affects, if at all, any of these effects of meth toxicity. And I'd like to thank Dr. Angelo for hosting me, Alan, my grad student, for teaching me so much in such a short period of time, and obviously Kristen for organizing this and making it a fulfilling and enjoyable summer. Thank you.